Today we're going to talk about, for the Journal Club for the Classics of Immunology, we're going to talk about a paper from 1953. We're going retrograde a little bit because last week we talked about 1957 and Sir McFarlane Burnet. But I wanted to finish off that whole tolerance and self, non-self recognition and that sort of thing first, because now we're going back to 1953. This really will round out the 50s, essentially. But as you know, I don't know whether you know it or not, but polio was a major problem. And it had been around for several hundred years, but always was not on an epidemic basis. And as time went on and towards the end of the 19th century, it started becoming more and more prevalent. And by the 1950s, 40s and 50s, it was a huge problem. And I happened to be a young tyke at that time, so I remember it distinctly. And everybody was really scared of polio, paralytic polio. Jonas Salk became Jesus Christ of vaccines at that stage of the game. So this was really his most complete paper that he published in 1953, and it was essentially a phase 1 trial of this vaccine in humans. So it was a dose finding and safety trial in humans. He had been doing experiments for at least a year or more, as we'll get into, in animals. But this was the first inoculation into humans. And so this is a little bit hairy, and it's an interesting story. So the title of the paper is The Studies in Human Subjects on Active Immunization Against Poliomyelitis. And it was published in the Journal of the American Medical Association, JAMA, which is one of the major clinical journals that's out there. So it was not obscure where he published this paper in 1953. And it's a pretty long paper. It's 17 pages long. And I found it to read now in 2021, turgid. And the prose was circuitous. And it wasn't sort of like the kind of scientific papers we're used to reading today, where every word is very carefully chosen, and because the publishers say, we only have so much space and we can't publish your paper, Dr. Smith, because you're too verbose, it's too long. But in this instance, he was very verbose, and it's hard to really read through the paper to figure out exactly what he's saying and what happened, what the data were, and what so forth. To give you a little background on Jonas Salk, he was born in 1914, and of course, Sir Peter Medawar was born in 1915. So they were obviously contemporaries. Salk was a Russian emigre. His parents were emigres. And so he was of this classic first generation American who was afforded all of the benefits of living in the United States of America. He was a really hardworking student. He went to college at the City College of New York, which at the time in the first half of the century, City College was very prestigious to get into. It was totally free. They really catered to the new immigrants, people. It was very competitive, difficult to get into. He went there when he was only 15, It was another one of those stories. And then when he graduated, he decided to go to medical school. He went to NYU Medical School. And then he went on from there to Mount Sinai in New York for his clinical training. And after that, in 1939, he went to the University of Michigan and did a postdoctoral fellowship with a fellow by the name of Thomas Francis. And at the time, Francis was studying influenza virus, sort of like Burnet. Everybody who was anybody was really studying influenza virus. And that was a very important influence on Salk, because Thomas Francis was experimenting with different ways to make flu vaccines in the early 40s. And then the war came on in 41 when they bombed Pearl Harbor. Jonas Salk was right in the thick of it. And as I said, when we talked about the flu virus, they didn't know how to attenuate it, like Pasteur had told them. that it wouldn't work unless they used a live attenuated virus. And so they did what it, it is what it is. You do the next best thing. And they tried to inactivate, kill the virus with formaldehyde, the flu virus, which they did. And that's what they used to then inoculate all of the recruits in World War II. And Jonas knew about all those experiments, how to do the inactivation with formaldehyde and then testing it afterwards and so forth. So ultimately, when the war finally got over and so forth, Jonas Salk was recruited to come to the University of Pittsburgh, where he arrived in 1948. And he basically decided to try to make a polio vaccine at that stage of the game. And he had been influenced by Thomas Francis and the flu story. And so he didn't even think twice about it. He decided to try to inactivate the polio virus. I mean, who would want to inject a live virus into a bunch of little kids and watch them get paralyzed? That wouldn't be such a good. So Jonas, he went off on this deal. And if you really want to read about the nitty gritty of this whole thing, there's a great book. It's called Patenting the Sun, and it's by Jane Smith. There's a lot of authors out there that are last name is Smith. It's a great book. She goes into all of the nitty gritty and the pressures and so forth, which were going on at the time. So in the introduction of this paper, Salk gives a little bit of the history. He said in this paper, he says that he's been already studying this to make a polio vaccine for more than a year, and studies in experimental animals, and particularly in monkeys. And the questions that he was trying to prove to himself, and then basically others, was whether or not it was possible to kill the virus, inactivate the virus, but still maintain its antigenicity or its immunogenicity. And that was an unknown question. And so that's what the raison d'etre of all these experiments actually was. And the purpose of this particular paper, as I said, he wanted to do the first experiments in the human. Now you can imagine that that's not an easy thing to do. And so, I mean, where are you going to get the volunteers? They're not going to line up off the street, that's for sure. In terms of the historical background of what was known about poliovirus when he was starting out, it was first discovered by Landsteiner, who was a fellow that we haven't really read his primary papers, but he basically was Mr. Immunology for the first half of the century because he was studying antibodies. Landsteiner was the first to describe poliovirus in 1909. And it wasn't until 1948, 49, that they discovered that there were 3 different immunological types, serotypes of poliovirus, type 1, 2, and 3. So it took 40 years to go from the initial discovery of the virus to the fact that this thing was pretty complicated, particularly immunologically. And so you could be infected with type 1 virus and then subsequently get infected with type 2 or 3. So just because you had one type did not make you necessarily immune to the other 2 types. Well, that's a little bit of a problem when you're trying to make a vaccine. And of course, now in the coronavirus days, we're all talking about these variants, which I wish they wouldn't use that word. I wish they'd use mutants so that people have to deal with what a mutant is and then find out about DNA and the structure and the genetic code and all these kinds of things that biologists today take for granted because we've grown up with all these things, but there's such a lag between what the biologists know about and what the common person knows about. It's unbelievable to us, to the biologists anyway. So it took 40 years to figure out that you needed to have, if you were going to make a vaccine, you needed to have all 3 of these types into your vaccine. The other thing that happened in 1949 was that, well, before that, the only way that the virus was propagated was the dogma was that it had to be the only tissue that it would grow in was nervous tissue, and so it was inoculated into the brains of mice and monkeys, and it was kept passaging that way. And the other thing that's important to understand is that this was before tissue culture. Nobody did tissue culture. It wasn't a discipline. And the reason it wasn't a discipline is that when people tried to put cells into culture or tissues into culture, mince up the mouse spleen or whatever and put it into culture. Within 24, 48 hours, it would be contaminated. It would be growing huge amounts of bacteria that were all over the place. And it was very difficult to keep those cultures germ free. So they would basically kill whatever cells that you were trying to grow, and it was just impossible. It just wouldn't work. Well, you know what happened during the war is that they started to use penicillin in big time, and they would take the urine of all the people getting the penicillin and re-extract the penicillin out of the urine and put it back in bottles and put it back into people because it was very precious stuff. But it worked really well, and particularly against what we call the pyogenic bacteria, with staphylococci and streptococci and so forth, that would cause really bad infections. So they for the first time in 1948, 49, they started using penicillin in their cultures. And at the same time, before that, streptomycin had been discovered as a very good antibiotic for the so-called gram-negative microbes, bacteria. And so the combination of penicillin and streptomycin was really the answer. And there was a group at Harvard, the head of the lab his name was Enders, and together with two young proteges of his, they tried to grow poliovirus in tissue culture, but not in nervous tissues. They tried a bunch of other kinds of tissues and were successful in growing the virus in several different monkey tissues. Ultimately, they settled on monkey kidney because it would form epithelial kinds of cells, and it would grow easily, and it would propagate the virus to high copy number, to high concentration. That was really a huge breakthrough. Both the combination that you could grow cells in culture and the fact that you could grow poliovirus in non-neural tissues. And that essentially, only 5 years later, after 1949, they had a paper. In 49, they had a paper out in Science that was just basically a letter, not even a full page, describing their experiments. And they went on to win the Nobel Prize in 1954. So this paper by Salk was published in 1953, but he didn't get the Nobel Prize. They gave it to Enders and Weller and Robbins in 1954. And I always thought that was an injustice, and I didn't think that was the right thing to do. But now I've gone back and I've read more about it, and I think educated myself. And so now I understand why they did it. And that is that the discovery that you could grow cells in tissue culture in the presence of penicillin and streptomycin was huge from a basic science standpoint. That was the beginning of tissue culture, number one, and the beginning of virology. Because virology before that had been, catch as catch can. You had to inoculate animals with it, and you really couldn't study that. It was, and so forth. This was a huge breakthrough, and that's why it turns out that there was on the Karolinska Institute, who gave the prize. They were really swayed by one of the virologists of the institute, who argued strongly that the most important discovery in this area wasn't the vaccine or whatever Salk was doing, but rather it was this basic science discovery, and so they gave it for that. And I think that's valid based on what's happened subsequently, and based on the impact the discovery of the tissue culture was, and so forth. Then I'll tell you what happened to Salk as time went on, and so he was okay. It's all right. I feel better about it now. So the question really became, once they realized that they didn't know how the infection happened in polio. The clinical manifestation of polio was the paralysis. So they knew that the nerves had been infected. They didn't know how the virus got from the outside to the inside and then into the nerves. They didn't know that it could circulate in the blood and secondarily infect the brain. But studies were done, and they found essentially that this is an enterotropic virus, meaning that it's transmitted through the intestine. So it's an oral fecal route if you can get your head around that. Contaminated water like swimming pools and so forth were a major problem and a major way that this virus was transmitted, particularly in the summertime throughout a community and so forth. So my mother wouldn't let me go swimming in the swimming pond. We didn't have a pool at that stage in the neighborhood. That was just fair boating. You didn't do that. But once they realized that the virus was coming in through the GI tract and then must have been going into the blood, they measured them and they looked for the virus in the blood. And one of the things that Enders and Weller and Robbins experiments allowed is that they could grow the virus, the tissue culture virus, and they could use that virus to look for antibody activity in the serum. And so now they had a test to look for the antibody activity. And they realized that the virus had to get to the brain or to the nervous system via the bloodstream. And that led them to do experiments in doing passive transfer of serum in animals and monkeys and so forth from monkeys that had been infected with the virus and recovered and were not, I don't know whether they were paralyzed or not, but anyway, they could test for antibody activity in the monkeys, and they could then passively transfer that serum into the serum from those in monkeys into fresh monkeys, and then challenge them with the virus, and they could show that the serum would prevent the infection into the blood. And that was another critical experiment that led them to think then if they could generate antibodies in a non-vaccine with a vaccine, that would work and they could prevent polio. Nowadays, I remember back when I was in school, in medical school, in our textbooks, they would say that 75% of the patients infected with polio were either asymptomatic or they only had sort of a GI flu kinds of symptoms. They didn't go on to become paralyzed, and like maybe 5 or 10% of the patients infected actually got paralyzed. Now, I've just been reading, they say that greater than 95% of the people infected with polio are asymptomatic. They don't have any symptoms. And so only about 4 or 5% have a GI flu kinds of syndrome, and less than 1% go on to having paralytic polio. So if they were having thousands of cases in the United States in the 40s and in the 50s of kids coming down with paralysis, and for the most part, it would affect the limbs of the lower legs, but it could move up, and if it got up as high as the neck, it would basically stop respiration through the diaphragm and also through the thoracic muscles and nerves and so forth. And so they invented the iron lung to put these people in, which they never got out of, essentially. It wasn't a good, fun time. The approaches that then Salk went on to try to do his active immunization. In 48, he first generated the formaldehyde fixed vaccines, and he showed that it would generate antibodies in the serum of monkeys. And it would protect those monkeys. They could challenge the monkeys by injecting the virus directly into the brain. The vaccine worked. It would protect the monkeys from paralysis, essentially. Then in around the same time, 49, 50, 51, there was several other people working in the business trying to make a vaccine. And it was, Salk just didn't come out of the blue. He had a lot of people breathing down his neck, essentially. And one of them was Hilary Koprowski, who was a Polish guy, aristocratic fellow who was at the Wistar Institute in Philadelphia. And I've met him. He was trying to make a live attenuated vaccine because that's how Pasteur said you had to do it, right? Now we know that's not true. So that's sort of the background in this. That's the introductory aspects of this paper. It goes on then into the materials and methods. He had to grow up huge quantities of the virus in order to make a batch of vaccine in order to have it and to do the quality control on the virus and get the right concentration and so forth and so on. And so he used tissue culture media at the time was pretty basic. It was essentially a balanced salt solution, but he used a tissue culture medium that's well known called TC 199, and he could grow the monkey kidney cells, green monkey kidneys cells in TC 199 without any serum, which was very important because it was to try to concentrate and purify the virus from the tissue culture medium. The serum was just be a hindrance in that whole. He grew up in tissue in roller culture bottles. And I don't know if you've ever seen a roller bottle. It's about this long. It's about this big around. And you put them on these rollers and they roll. And so that they're bathed in the fluid is sort of like, I don't know, 1/4 of the way up on the bottle when it's lying on its side. But as it rolls slowly, bathes the cells that are attached to the glass in the medium so that they don't die and they don't dry out and so forth and so on. And I used that when we first had to make a lot of T cell growth factor. He goes through, he gives data then in two different kinds of formats. One are in tables and the other one are in figures. And he needed to find out what concentration of virus. This would be the dose finding end of things. And so he would assay for the virus in different batches of his tissue culture media. And for the most part, he was getting pretty high concentrations of virus that could be then diluted out 1 to 10,000. And so that's what he used for most of his experiments when he started going first into monkeys and then into man. And in order to inactivate the virus, to try to inactivate the virus with formaldehyde, he mixed together the formaldehyde when you would buy it at the store would come in a very highly concentrated 37% formaldehyde, and then the other part being water. He would mix this together with his virus preps so that the final concentration of the formaldehyde was 0.4%. And then he would put the whole solution on ice, which he said was at 1 degree centigrade. If it's a refrigerator temperature, it's at 4 degrees centigrade. But anyway, he was at 1 degree centigrade, and he did it for different days. And he found that as time went on, we'll get into the safety tests, that he needed to have it incubated with the very dilute solution of formaldehyde for at least 10 days. To inactivate the virus. And after he did the formaldehyde treatment, then he would stop the reaction with sodium bisulfite, and then he would neutralize the acid concentration with sodium hydroxide. So this whole process, and this is what he borrowed from the flu business together with Thomas Francis. Then they would go on and they would take these different inactivation situations and they would do safety tests. And the safety tests, he did 2 different kinds of safety tests. One was to inoculate into the brains of monkeys, intracerebral inoculation of the things to see what would happen. And the other one was in tissue culture because after Enders had shown what would go on, if you incubated poliovirus with the kidney cells for any length of time, ultimately the virus would kill the cells, and you could you could see that under the microscope. So if you diluted out the virus and then looked every day, at the highest concentration of virus, you'd see that all the cells are gone, and then as they diluted it out, it would that whole what they call the cytopathic effect would get diluted out too. Of course, if you were successful in killing the virus, it wouldn't cause any cytopathic effect. And so those are the 2 different safety tests that he used. And he tried 2 different kinds of preparations with the virus to see whether or not he could generate antibodies in monkeys and then in humans. And one of them was he would just take the virus coming from the inactivation process and inoculate a very small part directly into the skin, which what we call intradermal injection. And when you do that, I don't know if you've ever had a test for TB, a PPD test. What you do is you stick a very small needle right into the skin, but not underneath the skin. That would be subcutaneous. This would be into the intracutaneous, into the skin. And when you inject about 100 microliters or so, you can raise a little bleb. It takes a couple of 3 hours to sort of dissipate and diffuse into the tissues. So that's intradermal injection of an aqueous solution. And then the other thing he did, and this was also taken from the flu business, he would use incomplete Freund's adjuvant. Incomplete Freund's adjuvant is everything except to kill TB bugs. So you take your vaccine or your antigen in an aqueous solution, mix it together with what they called aerosol A, which was a detergent essentially, and then mix that together with mineral oil, and then shake it up a lot. And that would then emulsify the aqueous solution into tiny little bubbles, tiny little droplets into the mineral oil. And I don't think anybody to this day has explained to me why this mineral oil and why this Freund's adjuvant is such a magic formula. But this is an example of how things start and how things go in the process. They found that if they inoculated that either intradermally or subcutaneously, that there would be a pretty severe inflammatory reaction, red hot, swollen, and sore. So they basically stuck it in deeper into the muscle, intramuscularly. And the whole idea in immunology has been passed down from one generation to the next, that the skin is a good recipient for an immunization. But I think that's a carryover from the days of Edward Jenner, when the skin was great. If you put your vaccine into the skin, and then you can just watch the skin. And if it gets inflamed, then you've got a reaction. And they would call that a take. And they did the same thing with polio. And you could see that there was something happening there. But with the oil, the inflammatory reaction was a little bit much. And so they put it into the muscle. And the muscle is all I think what really is the best thing to do is, if you've got a virus, for example, or a microbe that is transmitted through the respiratory tract, you could stick it in IV. You stick it in IV, it goes through the venous system into the right heart, and the right heart pumps it all the way throughout the lungs. That's where you want it. Anyway, I guess that it's off into a tangent right this instant, but the muscle is also good because muscle is highly vascular, because there's a lot of blood vessels in muscle, because they're bringing oxygen and taking away lactic acid and so forth, so we can all move around. And the tests that they used for the serum that they would get from these experiments, they used Ender's tissue culture cytopathic effect with serial dilutions, and they would always include, and Salk goes into detail about this in his paper, the pre-immunization sera as well as the post-immunization sera at different time intervals afterwards, and so that they would be in the same experiment to find out what the concentration of the antibody was in the serum. After they did monkeys, and they were pretty pleased with themselves because the monkeys really gave them a lot of information. And of course, with coronavirus, they haven't told the lay public about all of the animal experiments they've done in the past with all these vaccines that they've been talking about and so forth. And now they're only talking about the so-called phase 3 trials, where they're taking thousands of people and injecting some people with the vaccine, half of them, and another half with just saline. And then they wait to see if they get infected or not, and whether they get symptomatic or not. So for the first dose-finding safety studies, the phase 1 studies, and that's what this paper is about. Salk, he was in Pittsburgh, and there were schools, boarding schools for crippled children in the suburbs of Pittsburgh. And he used subjects from 2 of these schools. One was the Watson Home for Crippled Children, and the other one was the Polk State School in Polk, Pennsylvania. And he went into there and he injected 98 subjects from the Watson School and 63 patients or subjects from the Polk School. And of course, most of these kids had already been exposed to polio. They already had antibodies. So the experiment that he wanted to do, and that sort of gave him a little bit of justification that he wasn't going to make these kids sick all over again. And he'd done these safety studies so that he felt that he was on, that they didn't kill the monkeys. Hopefully they wouldn't kill the kids either. Most of these kids already had antibodies, and by this time they could test for the type specific antibodies. And so they knew who had what. Those were the trials that they then set up that they talk about in this paper. What the purpose of all of the experiments was is to see if the vaccine would make the antibody concentration go up in the serum of these kids. They first did only the intradermal inoculations of type 2 poliovirus, and they got antibodies out of that, and they were happy with that. But then they did later experiments, and they found that they didn't always get antibodies to type 1 and type 3, and so they went to the incomplete Freund's adjuvant with the emulsion and then the oil and intramuscular immunization. Out of that 161 subjects from these two different schools, they had no significant adverse events, no polio, no paralysis, and nothing really untoward at all, essentially. And so that was good. Of the people that they injected with the incomplete Freund's adjuvant vaccine, and they didn't have any severe local reactions, and that was good. The other thing they didn't know was, well, if they got antibodies after they gave the vaccine, how long is it going to last? And at the time that they did this report, the longest time that they had allowed to lapse was 4.5 months. So they knew that the antibody levels would go at least 4.5 months. They reported a bunch of experiments with monkeys whereby they did the formaldehyde treatment for less than 10 days and then longer than 10 days. 10 days seemed to be the cutoff period, and they could get antibodies as long as they used the incomplete Freund's adjuvant, they could get antibodies to all 3 types. So that's the end of the data. And in the summary and conclusions of this paper, they said, okay, we can make antibodies to all 3 types. We can get levels of antibodies that are in the circulation that are equivalent to the levels of antibodies in people who have suffered paralytic polio. And that was also good. And finally, they decided that they really, on the basis of this phase 1 trial, that they had to go on to phase 2 and phase 3 and inoculate lots and lots and lots of people. And so subsequently, in 1954, funded by the March of Dimes and the Infantile Paralysis Society, they did a double-blinded control trial all across the country. And Thomas Francis organized that and essentially helped Salk do it. And they ultimately immunized 1.8 million within a year, essentially. And so if you think about that today, there's been so much concern on the part of the populace of the safety of the coronavirus vaccine. But this is where we came from in the 50s, and this is where we are 60, 70 years later. And during that time, between the 160 kids in the phase 1 trial, and they just jumped right into a 1.8 million kids all over the country, and everybody lined up. I mean, people were so scared of this thing that they were not going to have their kid inoculated. As they say, the rest is history. Now, there's some follow-up of that whole thing. One of Salk's, and anyway, he ended up on the front page of Time magazine. He was a hero, and he became a household word, essentially, name. That was 1955 when I think they announced the trials from 1950 begun in 1954. But there was another guy that was trying to do what Pasteur told him he ought to do, and that was make a live attenuated vaccine. So that was Sabin. I think it was Arnold Sabin. So in 1961, Sabin announced that he had his live attenuated vaccine, number 1. Number 2, it was an oral polio vaccine because, it was an enteric virus. And so the virus would be just little drops, a couple of drops. And I forgot to say, I got the Salk vaccine, and then in 1955 and 54-5. And then when Sabin came out with the Sabin oral polio vaccine, I got that too. The Sabin vaccine then, because, well, I forgot to say, Jonas Salk was at the University of Pittsburgh, which was not necessarily the academic mecca in those days. Certainly not the way it is now. But Sabin was from the Rockefeller Institute, Rockefeller University in New York City, and he was well connected with all of the glitterati in American science. And because it was easy to administer and so forth, it basically did away with the Salk vaccine. And so everybody switched to the Sabin vaccine. As time went on, there were cases of paralytic polio that were cropping up. It was used all over the world. And cases of paralytic polio were cropping up as a consequence of the Sabin live attenuated, only partially attenuated vaccine. And in 1999, almost 40 years later, the American Academy of Pediatrics and all the other august institutions switched back. And so nowadays, children are inoculated or injected with Salk vaccine first. And I think depending on how it goes, they might have to have 2 different or 3 different doses to make sure all the types raise antibodies. But then when they get older, they start when they're 2 years old. And when they get older, then they get the Sabin vaccine is sort of a final booster on the whole thing, which I thought that story was very ironic. The other thing about the Salk story is that, they announced the Nobel Prize in 1954, and they didn't give it to Salk. They gave it to Enders and his crowd. And so there was a lot of people that thought that Salk ought to be recognized and so forth for his thing. And the March of Dimes, the Infantile Paralysis Foundation funded an institute for Salk, which they built on a bluff overlooking the Pacific Ocean in La Jolla, California, just a little bit north of San Diego. And it's quite something. If you ever get out there, or if you live out there, you should just check out the buildings from the Salk Institute. They're great. Francis Crick was recruited to come there after he won the Nobel Prize for the structure of DNA and so forth. And he was there for until he died, actually. And the scientists there, and I know several of them, are really good. And so that's a good part of the story. And I was there, I think in the 80s. And the students out there had invited me to give a lecture. So I went out and gave a lecture, and they took me to lunch in the cafeteria. And I was sitting there talking to 2 or 3 students, and who should show up but Jonas Salk, and came over to the table, and I met him and so forth. And it was fun. And I think I better stop there. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.